Dr. Raghu M.N., Associate Professor, Department of Pharmacology, to deliver his welcome speech.
Thank you, Arun. Good morning, everyone. I will take this privilege to welcome everyone. First of all, I would like to welcome our taking the blessings of our late R.L. Jalapa ji. First, I would like to welcome our Register Sir and the in charge Vice Chancellor, Dr. D.V.L.N. Prasad Sir. I welcome you, sir. Next, I would like to welcome our Dean and Principal, Sir, Dr. Prabhakar, who is our driving force in pulling and sustaining our college to our new heights. Next, I would like this as a privilege to welcome Dr. Krishnappa, Sir, our medical superintendent, who is with rich experience is helping us to move in a new direction, tackling all our problems. I welcome you, sir. Next, I would like to welcome Dr. Vinita Shankar, our Vice Principal, ma'am, who with a rich experience, who is driving our research to a new heights. I welcome you, ma'am. Finally, I will welcome all our delegates who have joined to this session. On this occasion, I would like to speak few words about Dr. Ramnath Chopra. Dr. Ramnath Chopra, an eminent pharmacologist, born on August 17, 1882 at Gujranwala, Punjab. After completing his early education in Jammu and Srinagar, he went to England in 1903. Later, he qualified to study medicine in Cambridge University, working with other eminent pharmacologists like W. Dixon. He later returned to India and he was appointed as a first professor of pharmacology at Calcutta School of Tropical Medicine. Later, he worked in Calcutta Medical College. Some of his outstanding contribution includes his work on Rolfia serpentina and other indigenous herbs of India. He later developed Chopra's antimony test for the diagnosis of leishmaniasis, which changed the treatment for the leishmaniasis. He compiled the first Indian pharmacopoeia in 1955. He was very instrumental in development of National Drug Research Institute in Lucknow, which is now called as CDRI. Many awards came in search for Ramnath Chopra, like Minto Gold Medal, Squibb Award, and Kaisari-Hind Gold Medal, and many more. To honor his contribution in the field of pharmacology, every year on August 17th is remembered as National Pharmacology Day. Hence, in view of this National Pharmacology Day, so we from our Department of Pharmacology, Sri Devaraja's Medical College, a constituent of Devaraja's Academy of Higher Education and Research, have arranged this guest lecture. Finally, I welcome one and all. Thank you.
Thank you, sir. Now, I request Dr. Bhuvana K., Professor and Head of Department of Pharmacology, to introduce our esteemed speaker of today's guest lecture. Ma'am.
Good morning, everyone. I'm honored and happy to introduce our guest speaker, Dr. Sharath B. Dr. Sharath B. pursued his doctoral degree from National Chemical Laboratory, Pune, with a research fellowship from the Council of Scientific and Industrial Research. He received Commonwealth split-side PhD scholarship to continue his work at the University of York Structural Biology Laboratory in the United Kingdom. He continued as a postdoctoral fellow in the Department of Pathology at the University of Washington in the United States. On his return to India, he joined Sri Devaraja's Academy of Higher Education and Research, Kolar, and then headed the Department of Cell Biology and Molecular Genetics. During his tenure here, he built a research program addressing the molecular basis of human diseases, which resulted in over 40 publications, patents, and extramural grants. He has guided 12 MPhil and PhD students. He has coordinated 5 national level programs funded by the Department of Health Research, Government of India, for training medical college faculty in the area of genetics. He is currently working as Deputy Director of IQAC at Enapoya, which is deemed to be University, Mangalore, and is accredited by A+ grade and 85th position in NIRF ranking. I welcome you, sir. Thank you. Over to you, sir.
Very good morning, madam. Can I share my slides?
Sir, give me a second, sir.
Yes. Now, shall I share from my side or?
Sir, you can, sir. I'm giving you credentials, sir. Yes, sir. You can now, sir.
Huh? Screen or will? Entire screen. Are my slides visible now?
Yes, sir. They are, sir.
Thank you. So, very good morning. First of all, am I audible? Hello.
Yes, Sharath, you're audible.
Very good morning. First of all, I thank the organizers, the Department of Pharmacology, Sri Devaraja's Medical College, Solar Constant Unit of Sri Devaraja's Academy of Higher Education and Research. Usually, whenever I had, it is my observation that whenever the Department of Pharmacology in that particular college organizes any seminar, first they try to see how it benefits the audience and then they decide the topic. So when I was requested to present this lecture, I was very honored and eagerly accepted. And one thing that I did not know was the basis for this pharmacology day. I thought there must be some conventional decision and they picked this particular date. And the preceding lecture given by the organizer, I came to know that it is not because just like the day has been picked, it because of a person who has contributed immensely to science, immensely to pharmacy, immensely to pharmacology and to health sciences as a whole. And I myself have some indirect links with the CDRI Lucknow, and today I came to know that he was one of the important person involved in setting up that institution. Otherwise, I did not know much about the foundations of CDRI Lucknow, which is the fountainhead of all pharmacy research in this country. With that, I would like to now go to my lecture. This today's topic is quite interesting. I have been associated with this topic directly, indirectly, in various ways, probably for the past 20 years. So when I was asked to talk on this, initially I felt it's a very simple topic and it is easy to talk on. But when I started going into it, I felt that it is actually a complicated topic, because usually when it comes to science, we only look at the techniques and some procedures and we start thinking, okay, this is all that is there to know about. But when I started looking at it in a holistic perspective, then I realized there are a lot more things, and I will try to do justice for whatever I have been able to find out. Now, these are the four areas that I will touch on. First, I will begin with the ethical issues involving animal research. This by itself is not the agenda for today's seminar, but I will just touch around to decide or highlight why alternatives are important. And then I will go to the scientific basis of animal study, because only when we understand these 2 points will we be able to understand what exactly is the need for alternatives. But most of my presentation will revolve around the techniques of the alternatives, and finally I will try to summarize what exactly is the current status of alternatives for animal experiments in today's world. So as I said, I have been associated with this topic for almost 20 years in some direct or indirect manner, and most of us, I think, have been involved in this, but mostly we come from non-science background, from public awareness background, and there are a lot of issues concerning use of animals. Whether somebody is scientist or not, we are all aware of some kind of a concern for using animals for research or for any other activities, and there are a lot of talk going on. So I'm not going to go into all those issues. So in all this background, when I looked at what I decided was, I will have to focus primarily on the professional view. That is, first of all, I will have to see why exactly we are trying to use animals, and is it justified or not? What are the strengths of animal research? What are the weaknesses of animal research? And therefore, in this scenario, what are the viable alternatives? So this is kind of a professional view I tried to assemble here and trying to keep biases of personal convictions away. Because when I try to look into literature, there are so much material which simply outright says so many techniques are available, so many procedures are there, still why people are using animal studies. So it looks a story is made out in the literature that animal studies are not at all required. People are simply using it. So I wanted to take a balanced view, taking both strength and weakness into account. Now, it's almost 75 years since the concept of alternatives for animal research has come in, and it was way back in around 1960s that the three principles of ours were introduced. I think most pharmacologists are aware of this. Reduce, replace, and refine. But the issue is how much we have been able to achieve all this, and to achieve all this, what we have done. So that is where the foundation for today's lecture is connected to. Now, why should we think of alternative or replacing animals with some other means of doing the experiment? There is a famous saying, an American saying, which says, if it is something is not broken, don't try to fix it. So we are already standardized all the animal experiments, they are doing well. So is there really a need to replace animals? So there is no problem in using animals. It is working out well. So then why should we think of alternatives? Now, first thing is ethics. So when I was fortunate enough to be on both human ethics committees and animal ethics committees, and the concept of ethics is usually misunderstood. We usually think ethics means we should, it's always aimed at stopping people from using animals or doing studies from studies with animals. So ethics always deals with whether it is necessary or unnecessary, whether it is justified or unjustified. That is what the whole exercise of the ethics committee and the ethics clearance focuses on. That is, we try to look whether we are doing a judicious work or not. All this is necessary because coming to quality and other aspects, because we believe in value-based society, we live in a value-based society. Therefore, as a college, as a university, we try to promote value-based education. So these alternatives for animals or animal ethics, these are not something by itself exclusively restricted to pharmacology department. Only pharmacologists have to worry about it. It is their problem. It is a bigger picture. It is a picture of the higher education as a whole, because we live in a value-based society, and the job of the colleges and the universities is to produce the citizens of tomorrow. Therefore, as a need, we promote value-based education. Therefore, it is everybody's responsibility to see what can be done about this particular issue. So the whole topic here, the basis for this topic is judicious use of any resource, be it animals or natural resource or human resource. Are we judicious in our use? Are we justified? Are we doing the right thing? And is it really useful for us? Is it really benefiting us? Should also be seen. The first thing I would like to address is it really justified to use animal study? Because today animal studies have become so strong that we simply take it for granted that animal studies are perfect. Now, whenever a research proposal comes to the ethics clearance, first point that is looked is scientific validity, not the ethical aspect of the proposal, the scientific validity, because unless something is not scientifically valid, it is not useful. And unless it is useful, it is not ethical to do such a study. So there itself, there is a point. If you see whether there is scientific validity or not, we can stop many of the animal studies. So we need to have a look first as to what exactly the animal studies are trying to give us and what they are not trying to answer, the strengths and weaknesses of the animal study. So then we will be able to decide what are the means for replacement, refinement, reduction, and then identify the alternatives. Now then, if you want to reduce, you also have to look for what are the alternative methods that are available to prove the same point. This is where the issue of alternatives and ethical aspect of animal use also comes in. Many times what happens is, we are used to animal studies because a setup is already available. So we simply take it for granted that we can do whatever experiments we want with animal models, and we simply do not ask the question Are there any alternatives? So that is also a point that we need to ask. For every experiment we are trying to do, first question that we have to ask is, okay, there are animal studies, but are there any non-animal alternatives to this? That point has to be asked. Now, when we ask that question, we also have to ask whether the alternatives are inferior to the animal studies, equivalent to the animal studies, or superior to the animal studies. If they are equivalent or if they are superior, we should always consider replacing the animal study. So when we look at all this, one confusion arises. We take it for granted that animal studies are useful, animal studies are meaningful, but we are not trying to address whether animal studies are required or not, or why we are trying to do animal studies. The next question that I would like to ask is, what information that we as scientists are trying to look for in animal studies? I can look at three broad areas where we are using animals today. First and foremost is academic research in colleges and universities, where mostly we do this for academic projects, student projects, teachers want promotion, so they do some projects or extramural projects, or there are some research institutes. They have some trust areas of research focused on some particular disease or some particular methodology to treat a disease. So they are trying to discover some new information, knowledge, or trying to develop some new techniques or gadgets. So they are all these are trying to aim at cause of a disease or treatment of a disease. And then comes the second part, which is toxicology testing. Toxicology testing again will involve some amount of academic research involving discovery and invention of new drugs. But I will come to this again in my later slide. You will see that toxicology testing is not something exclusively for pharmaceutical drugs. Industrial chemicals are also subjected to toxicology testing. And the third aspect again is drugs, coming mostly from the perspective of drug research institutes like CDRI in India and also all the pharmaceutical companies. What are the strengths of animal studies? Why we are so attached to animal studies? First and most important aspect of animal studies is experimental control. I don't think there are any other alternative methods where the control on the experiment is so enormous, because variable is a basic problem of all experimental design. So we need to control all the variables. And animal model is one model where controlling the variables is very easy. So that is why we always end up liking the animal models. Next one is uniformity of the subjects and treatment. This again is a difficult problem when we are dealing, say, with human beings or even with cell culture. Some alternative methods also has a problem when it comes to uniformity, whereas animal models gives a lot of control on ensuring uniformity of the subjects and in treatment protocols. And then comparing to human studies, here we do not have a very big problem of inclusion criteria, exclusion criteria. If you are looking at human studies, ensuring uniformity through exclusion is very tedious, laborious, and sometimes very expensive also. Enormous amounts of funds are required to do a battery of tests to decide whether a person will fit in this group or that group. And another important aspect is short duration, usually around 3 months, 6 months, most of the animal studies, at least one trial, one experiment will finish and people can move on to next experiment. And consequently, these animal studies have become ideal for student research projects. Most often, this is one of the biggest driver of usage of animals, because we can get the results very easily. And today, not only student projects, as NAC, NRF, wherever we hold meetings, first issue is there is no publication. So whenever this issue comes, immediately somehow the entire talk, discussion, plan, proposal, everything will channel itself towards animal studies, because they are quicker to do, easier to do, short duration, and also the greater subject control on the subjects. When you do all this, say, with human studies, there will be a lot of heterogeneity, and when you send your publications, journals will usually reject, saying there is no substantial homogeneity in these subjects. So there are a lot of unique strengths to animal studies, because of which we are kind of attached and we continue to use animal studies. But are there any weaknesses? We saw all the good points about animal studies, but we blindly think because of the strength we should use the animal studies. But are there any weaknesses in the animal studies? Now, when you look at cause of a disease, when you are using animals to understand the cause of a disease, you will see that there are a lot of weaknesses. Now, we induce disease in the animal model by various means, by some physical manipulation or by using some chemical damage or genetic manipulation. We induce disease models in the animals, and then we use those animal models to study. In molecular biology, we classify diseases as simple diseases and complex diseases. And if you see simple diseases, these are either infectious diseases. So the single agent there is the infectious agent, or single gene disorder. One particular gene is mutated and that particular mutation is responsible for the genetic disorder. For instance, thalassemia. The hemoglobin gene is mutated, only single gene is mutated, so that is the cause. So simple diseases are basically monofactorial diseases. Single agent is the causative agent. These are simple to study. And for that reason, animal model may be ideal to study here also. But today, as we all are already aware, most of the problems that we are studying in research are basically belonging to complex diseases. Complex diseases are mostly non-communicable diseases, and they are primarily multifactorial diseases. There is a genetic component, there is a non-genetic component, and the non-genetic component again involves a lot of factors, environmental factors, lifestyle factors, diet, nutrition, so many factors are involved. And even the genetic component, it is not a single gene, several genes are involved. That is why it is called polygenic disorders, or several genes are involved. So when you look at all this, the kind of diseases that we are studying today are complex diseases, and animal models are simply not the ideal models to study. But despite all this, if you look at how much of research papers are being published, say on diabetes, so many papers are there on animal studies. When you ask the question, are animal models justified? Then the clear answer it is no. So that also on one side it says animal models may not be ideal for all the problems that we are dealing today, but also it says we have to explore alternatives which are more suited to answer the questions that we are trying to ask. So its alternative is not only for the purpose of ethics that we have to be compassionate, we should also try to see what is the validity of the results that we are trying to get. So there is a point where animal studies are not about ethics or no ethics. It is about whether it is actually meaningful, scientific or not. This is the point I am trying to press here. Now going to the next weakness of animal study. In the previous slide, I tried to see what we try to get when we try to use animals for cause of a disease. Here, cause-effect relationship. For instance, drugs. We try to treat an animal with some kind of an agent when we try to see what happens to the animal. Now, first thing that we have to notice is there is a great genetic dissimilarity between humans and the animal model. For example, if you take aflatoxin, this is a toxin released by a fungus which grows on peanuts. If there are peanuts left to rot for a long time, some kind of a blackish fungus will grow on it, and it produces a toxin called aflatoxin, and it is a carcinogenic agent. And what we know today is it is carcinogenic to rats, it is carcinogenic to humans, but it is not carcinogenic to mice. So this is one example which says that there is a lot of heterogeneity in response to drugs also. So when we have such a scenario, how do you say animal studies, the results that we get from animal studies are easily extrapolatable to human beings, and therefore the research is useful. So when you see some recent survey, so there was a review carried out somewhere in 2017, and some of the authors involved in that were the former director generals of ICMR, some of the leaders of science of this country. They sat together and they saw what was the status of drug discovery in this country in the last 2 decades. And they saw the Indian R&D sector did not produce any new chemical entity in the last 2 decades. For last 20 years, simply we have been spending money, we have been putting a lot of effort, we are publishing a lot of papers, probably some minor patents also, and yet we have not found any new chemical entity that can solve any, that can treat any disease. And the authors, they feel that the failure to find any new chemical entity is because of the unpredictable toxicity or the poor efficacy of all the drugs chemical entities they find because of excessive reliance on animal model. That was their conclusion. That means we come to a point today where animal model, using animal, not using animal, has to do more with scientific validity of the animal itself in trying to answer all our questions and not merely compassion or no compassion. That is the message I am trying to drive here. So going on, we see that the major demand for animal studies, animal experiments is mainly in the area of toxicity. So now we saw three areas. First area where we are using animal is academic research, and another one is therapeutic, checking therapeutic effect, and third one is toxicity. If you take all these three areas, you see that academic research, mostly it is because of convenience people are doing. There is no really big substantiation right now with the kind of studies, diseases that we are trying to address to do animal model for academic studies. So that leaves the other two areas. One is the toxicology testing and therapeutic testing. Therapeutic testing, ultimately, a substance has to work in the human beings. There is no much big point in doing elaborate research in animals. But so that leaves the main area where animals are being used and continue to being used, and where we may have to continue to use in the future also, is in the area of toxicity testing. Now, when we come to toxicity testing, we have to understand that toxicity testing is not something exclusively for pharmaceutical drugs. It can also deal with food additives, cosmetics, industrial chemicals. Today, we are surrounded with large number of chemicals. The computers that we touch, the table, the wall, the paints, varnishes, the clothes that we use, the toys that children play with, everything is containing some form of chemicals or not. And in addition to that, all the fertilizers, pesticides that are being used in agriculture, which ultimately ends up on our table as food. Everywhere chemicals are there. So today there is a huge demand for toxicology testing because we live in a highly complex, chemically rich environment. But when we come to discussing the role of animals in toxicity testing or role of animals in research, we mostly focus on drugs. But there is a bigger need here. That is why I said the demand is huge, and we have been doing this. And this is the area where most of the animals are actually sacrificed, and therefore, and it is likely that it will also continue in the future. So this is the area that we have to mainly focus on. So this comes to one important point: whether should we continue now that so many animals are being used for toxicity testing? Should we continue to use these animals? Should we test for toxicity at all? So when you look at the literature, it is such a poignant story that the emphasis for statutory testing of substances for toxicity began somewhere in the 40s. And the whole story began with major problems arising out of cough syrup. In the 1940s, there were death of more than 100 children because of some preparations of cough syrup. And again, that was because of some of the solvents that were used in cough syrup. And nearly 100 years later, when you see it, today also we are suffering from similar scenario. That is, despite of making all this toxicity testing such important, we are continuing with the problem. That means on one side, we have the problems of usage of animals, but on the other side, we have the need for toxicity testing. Just because things have been used so long, that doesn't mean that we have come to a point, we have proved everything to be non-toxic. We know what is non-toxic. We know what is toxic. So such a kind of a black and white division is not available. So even today, we have to rely exhaustively, extensively, comprehensively on toxicity testing. So there is a need for us to assess what is the role of the animal and what are the viable alternatives. There is a whole battery of toxicology tests. Almost these are the 12 common toxicology tests, and depending on the different types of substances, far more tests may be prescribed. Now, what I have tried to do here is try to see what are the alternatives that are available for some of this, if not all of them. So the first one, so I will not go into the each method. So what I have tried to see is common tests available, at least to few of them, and then I will come back to this slide again and try to summarize how much we have been able to answer this particular issue. So now I will try to look at some of the tests or the alternative methods that are available today, because that is the whole emphasis, to show that yes, there are some alternative, and then we will see whether these alternatives are really valid or not, really useful or not. Are they going to help us in reducing the usage of animals, are they going to help us in replacing the usage of animals? First and foremost experiment is the skin absorption experiment. So this is one model that is available today. This is an alternative method that is available for animal testing. So this is basically a diffusion cell experiment, and most of you would have looked at some experiment similar to this in our undergraduate classes itself in diffusion studies. So basically there are two chambers. So if you see at the top there is one chamber, at the bottom there is one chamber, and in between there is one membrane. The membrane here is nothing but a cadaverous skin, or it can be a viable skin also. It can be, the source can be anything. It can be an animal skin also. So it depends. So here, because again we are dealing with alternatives to animals, so one idea is that we can use cadaverous skin. So the whole the principle behind here is, so there are two chambers, one at the top, one at the bottom, and in between you keep the skin as a membrane, and you pass some liquid through it, and in that liquid, in that solvent, you dissolve whatever the substance you want to test for absorption. And if that substance is, the test substance is absorbed by the skin, then it will pass through the skin and it will reach the bottom chamber. So you can collect the effluent in the bottom chamber, and you can perform some analytical tests and check whether the substance present at the top chamber has reached the bottom chamber. If it has reached the bottom chamber, that means the skin is capable of absorbing that particular substance. So you can say that is toxic. So this is one simple experiment. This is one simple alternative that is available for testing skin absorption. Now, next toxicology testing, that is skin irritation. What we are trying to test here? We are trying to test here whether there is a corrosion in the skin, whether the skin is being, some of the layers, cells of the layer of the skin is being digested, dissolved, disrupted. So here there is an alternative which involves use of reconstructed human epidermis. This is a alternative that has come because of a vast amount of research that has happened in 2 decades in the area of tissue engineering. So I will little bit later on talk about what is this reconstructed human epidermis. Basically, this is a in vitro cultured keratinocytes. Skin cells are cultured under in vitro conditions in the laboratory, and this is grown basically on a filter, and you can see in this left image here in a small cell culture dish, keratinocytes are grown in a filter. So why it is grown in a filter? Because cells, once you remove from their native condition, they will not form into tissue. So to give that framework, to give that skeleton, a polycarbonate filter is used. What is polycarbonate? It is a type of plastic. Our common plastic is polyethylene, and you might have seen polycarbonate. Some transparent plastics are basically bottles which are made. They are basically polycarbonate plastics. So polycarbonate mesh or filter is prepared, and in this filter the cells are grown. So this is basically an artificial skin. So it is reconstructed human epidermis. Reconstructed means what? Skin cells from a living human being is obtained and it is used to construct an artificial skin. So it is a reconstructed artificial skin. It is in vitro reconstruction. Human keratinocytes are obtained from a skin sample. So skin sample is obtained. Some protease enzymes are used to digest the skin sample. So the cells become free, and the free cells, the keratinocytes, then they are used to form the reconstructed epidermis. They are cultured under in vitro conditions on a polycarbonate or a plastic filter at air liquid interface. This is one challenge when we deal with keratinocytes, because keratinocytes have those properties only when they are in contact with air. I will show a few more images later on. When we do cell culture, usually the cells are submerged or fully immersed in the cell culture medium. But keratinocytes require that the cells grow in the air liquid interface, and some arrangements have been made to provide such an atmosphere for this particular type of cultures. And this is basically a primary cell culture. In cell cultures, we have 2 types of cultures. One is called a primary cell culture and another one is called cell line. What is a primary cell culture? Means these are cultures established from cells that are derived directly from the human source. So every time you want to prepare these cultures, you need to have a sample of cell from a donor. Somebody has to give you a piece of tissue. They have to donate a piece of tissue. Then only you can prepare these cell cultures. So this is not a cell line. Cell line means once somebody donates a tissue, it can be made into a permanent cell line. They are mostly cancer cell lines and they can be preserved in liquid nitrogen. They can be preserved in cryopreservation. So they can be preserved indefinitely and they can be grown in the lab also indefinitely. So any number of times you can use it. But this is not such a type of a cell culture. This is a primary cell culture. It means every time you need to prepare an artificial skin, there is a need for donation of skin also. Somewhere there is the need to obtain the skin sample also. So this is the monolayer of skin cells that I have shown. So this is not the artificial skin. This is not the reconstructed human epidermis. What I am trying to show here is how keratinocytes grow in cell culture. This is the original method that people were trying to do in laboratories last decade or 2 decades. Since last 2 decades, on the left side you see there are keratinocytes, and here it may not be clear, but basically these keratinocytes are attached to the cell culture dish and they are actually submerged in the medium. So this is a problem of growing any human cells in the culture, that is they are submerged in the medium. But the problem is in skin cell culture, we need the cells to be present at the air liquid interface so that they develop the properties of the skin cells. And traditionally, you may ask why, what is so novel about this method? Traditionally, we had very little, we had a lot of difficulty in growing human cells in laboratory, in growing under in vitro condition, because all the human cells are mostly differentiated cells. Differentiated cells have very limited capacity to divide under in vitro condition. So if you take a piece of tissue, you can easily get some cells out of it, but it is very difficult to grow them in the laboratory because they are all differentiated and they have very limited capacity for cell division or mitosis. And whenever they did grow, they grow as monolayers, that is a single layer of cells. But for all clinical applications or for some kind of a toxicology testing, we need organs or we need tissues, not one single layer of cells. So that was one challenge that we had. And today we have come to a point where there is a mechanism has been evolved where monolayer, instead of growing the cells in one layer, there is a mechanism to grow all these cells in a plastic meshwork so that they can form a artificial tissue. Here is a cross section of one such artificial tissue, and you see on the left side, the panel B is the actual human skin. On the right side, there is the artificial skin, and you see that the layers that you find in a normal skin are mimicked here. And if you see at the bottom of the image, there is a polycarbonate membrane. So first of all, that membrane or a plastic sheet, in simple words to say, provides a solid surface on which the cells will grow. And what may not be visible here is that some loose meshwork is also present, and the cells have kind of used it as a skeleton, as a mesh to attach themselves and form multi-layered cells. And this particular artificial structure now becomes a artificial skin. So going ahead, you will see that there are advanced versions of artificial skin also. And this is again, all these advancements are made by various biotech companies. And if I have given all the credits and the sources of these technologies also, this is by a company called Episkin, because toxicology testing, as I showed you, it is not merely for drugs, it is for industrial chemicals. So there is a vast commercial application for all these technologies. So most of the research on these aspects are going mostly in biotech companies rather than in pharmacology institutions. And here, if you see, this is an advanced version. Here you also have dermis being formed. And here, in addition to using solid surfaces, collagen-based matrices are provided to produce an advanced version of artificial skin. In the previous example, you saw that it is on a plastic film or a plastic sheet or a plastic membrane that the artificial skin has been formed or reconstituted. Here, they have gone a step ahead and used collagen as the framework to help these cells to aggregate into forming a tissue, artificial tissue. And again, you will see that the middle panel, when you see, all the layers that we expect from a normal skin are present in the artificial skin. So going back, we use all this artificial skin in a toxicology testing. That is the whole objective. How do we use it to solve the problem of creating an alternative for animal testing? So again, once you form a artificial skin, you here, if you see on the right side, you see there's a bigger petri dish, and inside that there is a smaller petri dish. Inside the petri dish, at the bottom, there is a blue layer, which is nothing but the artificial skin, the reconstructed human epidermis. And on top of that epidermis, human reconstructed epidermis, you can place your test compound. And if that's then if that first of all, if it is able to absorb, it will pass through the skin layer and it will reach the bottom layer. First of all, that will tell whether the test compound is able to be absorbed by the reconstructed epidermis or not. And then in the next step, it will, you can take that particular sample of reconstructed epidermis and carry out microscopic evaluation and see whether there is any cellular damage or not. So from that, you will be able to tell whether there was actually corrosion in the cell layer or not. If there is skin irritation, there should be damage to the cell. If there is skin absorption, the substrate will simply enter the layer of skin and pass through the skin. If there is corrosion, then the skin cells will be damaged. And this here you can carry out microscopic evaluation, and then you will be able to check whether the given substance is toxic or not. But one particular drawback of this method is mild irritants cannot be tested by this method. So this is one problem which will keep on repeating in all the methods. That is, one category of toxic substances can be tested, but all categories of toxic substances may not be tested because skin is highly sensitive. Here, mild irritants may not be sensitive to produce any kind of observation, whereas strong substances, strongly toxic substances will produce some kind of an observation. So these methods are possible to identify strong toxic agents, but if they are mild, they will produce false negative results. So that is the limitation of this particular method. And in every method that I will speak later on, you will see this will be one common scenario. Going ahead, now we'll come to phototoxicity. So what does phototoxicity means? Whether a substance is absorbed by skin, and when the material that is absorbed by the skin is exposed to the UV light present in the sunlight, does it undergo some kind of a change? And because of the change, does it kill the cells or not? So phototoxicity is one of the common toxicology tests. And today there are cell culture alternatives for it. So here a particular cell line is used. This is not a primary cell culture. This is a cell line. That means cells have been made and it has been kept. So you can use this cell line. So it is called the BALPC 3TC cell line. It is basically a mouse embryonic cell line. From the mouse embryos, the embryos were disintegrated and cell lines were prepared, and cell lines are cryopreserved. So you can use these cell lines to test the given compound. So basically, you take this cell line and you expose your test substance for 1 hour, and then you let the cell lines grow in the dark. You let the cells grow in the dark for 24 hours, and then you check whether the cells are viable or not. If the given substance is toxic, then the cells will die if you expose it to light. Expose it to light. If they are not toxic, they will survive. So let us see some examples here. If you see the top panel here, you see the panel B. The test substance 0 to 200 means a range of concentration of the test substance has been given. So you do a titration of the test substance, and the whole experiment has been carried out in the triplicates, and you will see up to about 50 micromolar concentration. This particular component is not toxic, but after that it is toxic. How do you know it is toxic or not toxic? Because after 24 hours, all the cells here have been stained with a dye called neutral red. Neutral red is a vital dye. If the cells are alive, if the cells are viable, the cells will be stained with neutral red. If they are dead, if they are non-viable, they will not be stained with red. So the cells on the left side here, these cells are living, these cells are dead. these cells are viable, these cells are not viable, or the given substance was non-toxic in this range, but was toxic in this particular range. Now, the same experiment has been carried out in two particular setups. See, in the first setup, it is minus UV. That means this particular plate was not exposed to UV light, whereas this particular plate was exposed to UV light. And you see here, when it is exposed to UV light at around 10 micromolar concentration itself, the cells have started losing viability. It has become from pink, it has become palish. So at 15 itself, at a very small concentration, cell in this particular concentration itself, it is fully dead. Here, if you see 120 up to at 140 only, you are seeing completely dead wells. Whereas here at 15 itself, you are able to see. That means this particular compound that you have tested without UV light, with UV light, with a range of test substance using BALB-C 3TC cell lines, this particular compound is phototoxic. Like this, you will be able to prove. Like that, if you see another test substance has been tested, and you will see both without UV, with UV light, the cells are viable in all the ranges. 0 to 250 has been tested here. The cells are viable all the way from 0 to 250 concentration without UV light. In the presence of UV light also, 0 to 250, they are viable. That means the test compound in this panel D is not phototoxic, whereas the test compound in panel B is phototoxic. So this is one experiment you can use. Again, the same problem remains. What is strongly acting, you can test. What is mild, you will not be able to test in this particular method. Now going to the other, yet another toxicology testing, which is skin sensitization. So again, there is an animal test for this, but what is the alternative method for this one? So what is skin sensitization test? Basically, this test tries to check for whether there is an allergic response when a given test compound is exposed to comes in contact with the skin. Now there is a in vitro method for this. So basically, to summarize, this is basically an in vitro monocyte activation experiment. So you can collect monocytes and we can activate we can collect the monocytes from the human blood and we can treat it with the test compound and we can check whether the monocytes are activated or not. So I have represented it pictorially here. So you can prepare monocytes from the human blood samples. So you can take from a donor and you can isolate the monocytes and you can incubate those cells with your given test substance. And if it is allergic, if it is, then the monocytes will undergo activation. During activation, there will be a transformation. The monocytes will try to become dendritic cells. So there is a change in the profile of the cell. So this will be accompanied by changes in the expression of the surface markers. So there are two classical markers called CD86, CD54. So you will be already aware that one method that is available to test all the surface markers is flow cytometry. So when you expose the given monocyte sample to test substance, whether it is still as monocyte or whether it is converted into the dendritic cell or not, you can check by the presence of these surface markers through flow cytometry. So this is a simple method where you can look for sensitization without having to perform any complicated animal experiments. So that was a cell-based method. Now there is for sensitization, there is what is also called as a in chemico method. What is an in chemico method? In chemico method means basically it's an in vitro method using only a chemical reaction. Now cell cultures are also in vitro methods. In chemico method means we are not using any cells. We are trying to use some reagents. Basically, it's a type of a biochemical reaction that we are going to use here. So all these methods will finally focus on what is called as the adverse outcome pathway. So what is a pathway? We are aware of biochemical pathway which runs through the human system. Likewise, inside the cell, there are certain biochemical pathways. They are called as molecular pathways. Most of them are involving some proteins and some small molecules. And some pathways, and the role of these pathways is to activate the cellular response. Just like the way physiological pathways, biochemical pathways will activate the clinical response, these adverse outcome pathways are intracellular pathways which will activate the cellular events inside the cell. So they are basically the molecular initiating events. These are the pathways that will initiate the or trigger the cellular events, which ultimately underlie the clinical effect. The key events that are the key cellular events involved in adverse outcomes are at the cellular level. Covalent interaction with the skin proteins. First of all, the test substance has to interact with the skin. If it has to sensitize the skin, first it has to interact with the proteins of the skin cells. So if it is not going to interact with the skin proteins, then there is no way that particular substance is going to sensitize that particular cell. Then what happens to the keratinocytes? The events inside the keratinocytes. Once there is some changes in the keratinocytes will release some cytokines and some other molecular substances, or it may undergo apoptosis and some damages will be there. And all this will activate the peripheral cells. So first is contact with the skin proteins. With that, there will be some kind of events inside the keratinocytes. Once there are events inside the keratinocytes may release some cytokines, or keratinocytes may undergo necrosis, they may die, they may release some substance, and this will activate all the other cells present in the skin. So next will be the events that happen to the that happen inside the dendritic cells, and then the events that happens inside the lymphocytes. These are all the different cellular events that will happen during the process of skin sensitization. And all these together, and in all these events, when they are happening inside these particular cells, be it in the skin cell or in the dendritic cell or in the T lymphocyte, there are certain cellular molecular pathways, and these pathways are activated, and these pathways will eventually produce the adverse outcome at the cellular level. The adverse outcome at the cellular level will eventually underlie the clinical observation of skin sensitization. So how exactly do you do the skin sensitization test by in chemico method? So basically here the method that we have, I am only going to focus on covalent interaction with skin protein. What is covalent interaction? How you are going to do that? Now this is basically you are going to test for the capacity of the test substance to bind to the skin proteins. So to check whether a given substance has bound to the skin protein or not, you don't need a skin by itself. You do not need the skin cells also by itself. You just need the skin proteins, and you don't need the total proteins also. You just need some peptides or pieces of protein. So what this particular assay does is there are synthetic peptides that have been used, and these synthetic peptides contain either cysteine or lysine, and most of the interaction with the skin protein with the toxic agent is because of the interaction they have with the cysteine and lysine residues present in these proteins. So synthetic peptides are prepared containing cysteine and lysine. So you incubate or you treat the substance with the synthetic peptide for about 24 hours or overnight, and then you check whether your given test substance has bound to the given peptide or not. So that is the reason why this method is called direct peptide reactivity assay. So you are checking for reaction of your test peptide with the reaction of your test substance with the synthetic peptide. If it is bound, then how do you check it? Now, if it is bound to the peptide, the peptide will be lost. Whether the peptide is free or not, peptide is bound to the test substance or the peptide is free. If it is free, you can measure it by HPLC. I think most of you would be aware of what is HPLC. It is a very common method, and the common method to determine substances, peptides also in HPLC is by an analyze detector called UV detector. So you can carry out all these experiments in the microtiter plate, and you can analyze it by mass spec or a simple or a common HPLC also. So you can check whether the peptide is, basically you check whether the peptide is free or not. If the peptide is free, that means it is not bound to the test substance. If it is not bound, that means the given test substance is not toxic. It is not toxic with respect to skin sensitization. It is not capable of inducing skin sensitization. So this is a preliminary screening test, which can be, the reason why it is shown as a microtiter plate here is that large number of substances can be screened here. When you do animal studies, first of all, you can do only few animal studies, and then you have to do them in triplicates or multiples. So there is a challenge of number of animals that you can use. But here you see, you can have a high throughput screening also. So instead of testing one or two substances, a large number of substances in large number of concentrations and forms and combinations, they can be screened in a high throughput manner. So that is the strength of this particular method. That is the main in chemico method that is available for covalent interaction with skin proteins. Today, there are not that many methods available for in chemico methods available for understanding whether a given substance is going to produce any damage inside, initiate any event inside the keratinocytes or dendritic cells or T lymphocytes. Here I can only give a preliminary indication, just like the way there is something called Human Genome Project 20 years ago, they did Proteome Project. So many projects have come, huge, gigantic projects. Likewise, there is an effort to carry out something called as a Human Toxicome Project. Basically, what people are trying to do is, if the common categories of toxic substance, when you expose them to cells, what are the common pathways that are going to be activated inside a cell that is being understood? Today, the results are not fully clear. There is no result to say these are the 10 pathways or these are the 100 pathways that are going to be activated inside a cell when this particular category or a class of toxic agent is exposed to a cell. But if that is made possible, then we will be, without having to test in animals, we will be able to look at the chemical and we will be able to carry out some in chemico methods. So by looking at one of the starting agent, the starting chemical substance or the starting molecule in this pathway, we can carry out interaction experiment in that pathway, the starting point of that pathway, and check whether this pathway will be initiated by this particular compound or not. But at this stage, most of them are futuristic in terms of technologies. They are not currently available. Whereas covalent interaction with skin protein, this is a test method which is already available, and to some extent, it is also being used. That is why I only focused on this, but more are in the pipeline. Now, the next alternative method is for the ocular irritation test. So there is an in vitro method here. So I think this is one of the, I think there is something to be carried out in the animal model. It is carried out in rabbits, in the rabbit eye, which is a very painful method and also dreadful method. But today there are some alternatives, and this method is called, the alternative in vitro method is called in vitro macromolecular test. So basically, a transparent macromolecular matrix is formed. Now, before I go into all this, I will have to convince you what exactly is the meaning of the word macromolecular. So very simple, we have all studied biochemistry. Molecules, biochemistry is made up of molecules, large molecules. What are the large molecules? Proteins, lipids, and nucleic acids, carbohydrates. These are large molecules because they are mainly polymers. When I say carbohydrates, also large carbohydrates, polysaccharides. Proteins, they are polymers. Glycoproteins are nothing but type of proteins, proteins plus carbohydrates. Carbohydrates, basically, I am talking about polysaccharides. They are again large polymers. Lipids, they are also polymers. So all these polymers, these polymeric substances are called macromolecular substances. So all these macromolecular substances can be pulled together, can be put together to form a macromolecular matrix. So this is basic. In simple words, you can say a gel. You can say a transparent gel. In simple words, it is nothing but a transparent gel made up of various macromolecules that you see in the human eye. So basically, this macromolecular assembly will mimic a highly ordered structure of the cornea. It is not the cornea by itself. It is not a reconstructed cornea. It is a mimic. So we are trying to assemble a large number of polymers into a type of a gel, and one picture of it is given here. So you can prepare this macromolecular model, and into that macromolecular matrix, you can treat your test substance. So you will see, you will put the test substance in the macromolecular matrix for about 24 hours, and then you will see whether the macromolecular substance remains stable or it becomes turbid. What is the meaning of turbid? It will start to disintegrate. So basically it is reflecting the opacity that will be developed in the cornea if the given substance is capable of inducing ocular irritation. Now, what will happen if you're, let us say, how do you read a positive test? So you have prepared your macromolecular matrix, you'll expose it to the test substance, and let us say the test substance will degrade the matrix, so the matrix will become turbid, it will become cloudy, it will become opaque. So how do you measure this? So turbidity we measure by using light scattering experiment. Light scattering experiment, or basically it is called turbidometry. In biochemical analysis, you might have heard of some methods called nephelometry for antibodies that are commonly used. For C-reactive protein, it is used. So these are all turbidometry and nephelometry are different types of light scattering methods. So a test substance is used. Let us assume there is a test substance which is mixed with the macromolecular matrix. If it is capable of degrading the macromolecular matrix, then the test substance will become, your matrix will become degraded and it will become cloudy. It will become turbid, and you can do light scattering. Basically, light is being thrown on the matrix here. The light is scattered, and the scattered light is measured by a optical detector. So depending on whether you measure it in line with the light source or at some angle with the light source, the experiment will be called turbidometry or nephelometry. And today, all the large macromolecular tests that are conducted in the biochemical lab, like antibodies and CRP proteins, they are all, some of them are based on nephelometry or a type of turbidometry test. So to summarize, this is basically an artificial macromolecular matrix, which if it is degraded by the test substance, then we can classify it as toxic. Toxic with respect to ocular irritation, or we can classify it as an ocular irritant. Now going ahead, there is another method which is an in vitro method, but this is a cellular method. The previous one was an in vitro method. Again, it is an in chemico method. No cells are used there. In vitro, in chemico method using some assembly of chemicals. Here we have we have an in vitro method, which is a cellular method. So like in the previous case of skin epithelium, there is today a possibility of reconstructed human cornea-like epithelium. So again, this is a primary cell culture that has been assembled here. So you can see at the right corner, top right corner here, this is a cell culture dish, what is called as a 6-well plate, because there are 6 wells in the plate. And in the middle, there is a septa, or a small tub or a small cup is there. At the bottom of the cup, that particular cornea-like epithelium has been grown. And that pink fluid is nothing but the cell culture medium. And you can look at the panel C and D at the bottom, and you see the panel C, you will see that there is a microporous membrane or a solid surface or a plastic surface. In simple words, it's a type of a synthetic plastic. It's a plastic material which provides the solid support or the base under which the corneal cells will grow and form a multi-layered tissue. And this has been compared to a normal corneal cross section also, and you see whatever layers you see in a normal in a natural corneal tissue is also present in the artificial or the reconstructed corneal tissue also. And like in the previous experiment, you can have this these corneal layers will be formed in will be reconstructed inside a small tub. It is called a septa, and that will be kept in a bigger dish. And whatever substance you have, you place it on top of the cornea-like epithelium, and then you can carry out the microscopic evaluation to check whether the epithelium is intact or not. If it is intact, that means the given substance is not going to corrode the ocular epithelium. If it is not intact, that means the given substance is capable of corroding the optical ocular epithelium, and you can classify the given substance as irritant. So now we'll go to the next test. So this is basically a test to check for chromosomal damage. So this is basically called a mammalian cell micronucleus test. So before I go to the test itself, I would like to tell you what exactly is a micronucleus. So all of you know what is a nucleus. Every cell has a nucleus except the uninucleated cells. In the nucleus, sometimes you see small nuclear-like nuclear structures, they are called micronucleus. This is not a normal observation. Sometimes you see small nucleus-like structures in addition to the normal regular nucleus. So you see here in the middle of the entire blue tinged area is the whole cell. In the middle there is a nucleus, and in the periphery you see there are 3 small micronucleus. How they are formed? They are basically extranuclear structures formed in the cell. How they are formed? A small broken piece of a chromosome. Yes, because of DNA breakage due to damage to the DNA. If there is a damage to the DNA, then what happens? Chromosome will break. If the chromosome is broken, then it cannot be thrown out of the cell, isn't it? There is no garbage disposal mechanism just like that inside the cell. So cell will retain the broken DNA by forming a small micronucleus, or whole chromosome that fail to segregate to a daughter cell. So you might have heard of genetic disorders, euploidy, aneuploidy, all those things. So aneuploidy means what? Either one chromosome is gained or one chromosome is lost. So if one chromosome is gained, what will happen to that extra chromosome? That extra chromosome may undergo, sometimes may not remain intact, may undergo, it may become inactivated and can form a micronucleus. Okay, so both of them, why do you get a piece of chromosome? The piece of chromosome arises because there is a DNA damage. If there is DNA damage, chromosome is broken into pieces, the piece will form micronucleus. If there is a spindle poison during mitosis, spindle apparatus is formed. What is the role of spindle apparatus? The role of the spindle apparatus is to ensure that equal number of chromosomes are segregated for the daughter cells. Equal number should be there in both the cells. But what happens if there is a damage to the spindle apparatus itself? Then some unequal number of chromosomes will go. So basically, this is a test which will check whether there is the given substance is capable of doing any DNA breakage, or is it a spindle poison? Because if there is a DNA breakage, chromosome will break, chromosome will form micronucleus. If the given substance is a spindle poison, spindle apparatus will collapse. Spindle apparatus are like railway lines. So the chromosomes cannot go to the daughter cell. Both the chromosome will end up in, instead of two chromosomes going to one each, both will end up in one daughter cell. So there that extra chromosome will undergo inactivation and form the micronucleus. So this basically the mammalian cell micronucleus test is a test for chromosomal damage, or what are called as genotoxic agents. So you need not do all these tests in animals. So a simple cellular assay is also capable of identifying whether a given substance is genotoxic or not. Going to the next test. So this is basically a test to check gene mutations. No, the substances that cause gene mutations are called mutagens. Now, what is the difference here? Substance which can cause mutations are called mutations. Substances which cause chromosomal damage are called genotoxic agents. Genotoxins means they cause large scale, huge damages. These are more toxic than mutagens. Mutagens means they will not cause any breakage of DNA, they will not cause any breakage of chromosomes, but they will cause changes in the nucleotide sequence. So instead of coding for one protein, it may code for another protein, or it may not code for any protein. So these are all milder than genotoxic agents, but it can still affect whether the gene will function or not. And as a consequence, it can simply switch off a gene. So here, again, so many tests are available here. I will not review all the tests. For the sake of illustration, I will tell you one test which is called as the Eames test. And this is based on using bacteria. This is a bacterial test. Again, here also cellular tests are also available, because most of the experiment alternatives that I am talking uses only the cellular alternatives. So here for a change, I thought I will restrict my discussion to bacterial test. So this bacterial test is called as a Eames test, based on the founder of this particular test. And the principle behind here is reversion mutation. Here in this, I will tell you what is reversion mutation. First of all, which bacteria used? Here bacterial strains of Salmonella typhimurium will be used. And this particular bacterium has various strains called as auxotrophic strains. And these strains of this particular bacterium are used. And what are these auxotrophic strains? Basically, these strains are containing some mutations. There are already some mutations in this particular auxotrophic strains of Salmonella typhimurium. So the starting material is a Salmonella bacteria containing some naturally occurring mutations. They are already present. And the given test substance, if it is a mutation, if it is a mutagen, the given test substance is capable of inducing mutation, then it is causing mutation on a mutation, then it is called a reversion mutation. That is, mutagen causes mutation of a pre-existing mutation. Mutation of a mutation is called reversion mutation. What happens because of reversion mutation? A capacity which was lost because of the mutation is now being restored. Reversion mutation results in the restoration. It results in the restoration of the original capacity. Now, here in this particular Salmonella strain, auxotrophic strain, auxotrophic means something to do with bacterial nutrition. And this particular bacterial strain has a mutation in histidine biosynthesis pathway. Because of this, these strains cannot grow on any bacterial culture medium that do not contain histidine. So the bacterial culture medium has to be supplemented with histidine because they cannot produce histidine on their own. So if a given test substance causes mutation inside the mutation, that is mutation of a mutation, then the bacterial strain will now be capable of growing in a culture medium that is lacking histidine. So we are looking for restoration of the capacity to synthesize histidine or restoration of the capacity to grow on a medium without requiring any supplementation of histidine. So I will just show the same thing with an experimental flow. So here there are two tubes, an experimental tube and a control tube. And you see here it is treated with the test substance and both of them are poured on the bacterial cell culture plate. Here the bacteria are struggling to grow. That doesn't mean the results are zero. There are still few, some 2 colonies have grown, but here large number of colonies have grown. That means the given test substance was kept, and this particular medium here is histidine selective medium. That means in this particular medium, histidine is not provided. If you provide histidine, this bacteria will grow. If you do not provide this histidine, this bacteria will not, this particular strain of the bacteria will not grow. It will not grow because histidine's biosynthesis gene is mutated. Now, if that mutation undergoes mutation, there is a restoration of the mutation. Now that bacteria will now regain the capacity to grow in a medium lacking histidine. So here it is growing, large number of colonies are formed. And you see here in the control plate, very few colonies are formed, but that doesn't mean no colonies are formed. That means there is always a background mutation, whether you treat with mutagen or not. This is one molecular principle. There is always some background mutation. Adding a mutagenic agent will only increase the frequency of mutation. The chances of mutation will increase. So this is a bacterial test. This is a well-known classical test in molecular genetics for testing mutagens. So now we'll come to the next method, which is cell transformation assay. This is basically a test to look for carcinogenicity. We want to check whether a given substance is carcinogen or not, whether it can cause cancer or not. Again, here we are using the biopsy 3TC cell lines, which is nothing but the mouse embryonic cell lines. And here we do what is called as a colony formation on soft agar. Cell culture, normally we grow in liquid media. I already showed you in one of the previous slides. There was a pink fluid. When you tilted the plate, that fluid was also tilting back. So for simplicity, I'll go back and show you. If you see here, when you tilt the plate, the liquid is also tilting down. That is because this is a liquid medium. Cell cultures are always grown in liquid media. Because cells grow only in liquid medium under in vitro condition. However, and those are all normal primary cell cultures, means cells obtained directly from the human beings and they are normal cells. But one property of the cancer cell is they can grow in soft agar. Just like agarose that we use in bacterial laboratories or bacterial cell culture, there is agar, which is a soft agar made of agarose, which is little more refined than agar. And cancer cells can grow in soft agar, whereas normal human cells or normal mammalian cells will not grow in soft agar. That is because these cancer cells, they have the capability of growing by themselves, and they have basically the capacity to grow without being attached to anything. If you saw all the experiments that I showed you about reconstructed corneal epithelium, reconstructed skin epithelium, there is a solid surface that was provided for the cells to go and attach and form the tissue, because they are all normal cells. Whereas cancer cells, they need not attach. That is one of the reasons for metastasis. Cancer cells have the capacity to grow without being attached to any solid surface. So that particular property is being tested here. Soft agar is made, and that soft agar is mixed with the the cell culture. First, the cell culture and this test substance are incubated with each other for 24 hours, and then they are grown in soft agar. Normal cell will not grow in soft agar. If you see here in the right panel, the right plate, there is no growth at all. Whereas the left plate, you see the cells are grown. It is stained in blue color. So this is grown because that means they are capable of growing inside a gel. Agar is what it says, gel. Gel is not a solid surface. Cells cannot grow in a gel. Gel is nothing but a liquid-like substance. It is not a solid substance. It is not a solid substance. So cells will always require solid surface to attach and grow. Cells will always attach and grow. Whereas here, these cells are on the left side, these cells are growing without attaching to the cells, without need for any solid surface to attach. That means these cells have undergone transformation, and the difference between this cell culture and this culture, both of them started with the same cell sample, but this particular sample was treated with a test substance. That means this particular test substance is a carcinogen capable of causing transformation of the given cell culture. So you can classify this particular substance as carcinogen. So I have tried to present all the methods that are commonly available. There are far more methods for some of the rarer tests also available. I have not tried to review all the methods here, but I have tried to review some of the common methods available, and some of the common methods are such that most molecular labs or cell culture, cell biology labs will be able to set up such kind of experiments. I have tried to present here. So what are the, now to summarize the application, the whole idea of all this method was not the novelty of these methods, but whether they are useful in terms of becoming an alternative for animal experiment 1, and then 2, replace the animal, and 2, provide meaningful, scientifically valid results. Now that is a point that we have to check. Now the major limitation of all these methods is most of these methods are validated for a few classes of toxic substances, but not for all the classes of toxic substances. So they are not universal tests. So you cannot simply assume that every class of toxic agent has been can be tested by this particular test. So if a given substance is positive in this test, yes, you can say it is positive. But if it is negative, you cannot say it is negative, because we are not clear whether the test is validated for that particular category of toxic substance or not. And the next major problem with all this in vitro method is the effect of drug and xenobiotic metabolism cannot be simulated. Whenever we do an animal study, the drug is ingested or the drug enters the skin, it undergoes metabolism through various enzymes, be it a drug or even any other substance, even a drug. Basically, all these pathways are called as xenobiotics pathways, because drug is also a foreign substance. All industrial chemicals are foreign substances. When these substances enter the human body, there are certain enzymes within the human body which are evolved basically to inactivate these drugs, and so that they can be eliminated. Basically, they are present as a detoxification mechanism to degrade the given foreign chemical and excrete it out of the system. Now, this particular xenobiotic metabolism, because of the total physiological metabolism, cannot be simulated under in vitro condition, and this is the major problem of the limitation of all the methods that I showed you. And then, as a result, if you see most of the methods that I showed you, the artificial skin, the artificial cornea, majority of the alternatives that are available mostly deal with surface active agents, because if the drugs have to reach some internal organ, then definitely they will undergo xenobiotic metabolism. So unless you are clear what exactly is the final product of that particular drug or the industrial chemical that will reach the target organ or the target tissue, you will not be able to use that particular form of the test compound. So you will be able, you will end up getting false negative results for drugs that are not or substances that are not immediately acting on the surface organs or tissues. So I come to the end of my presentation. To summarize, overall, we can tell there are some alternatives that are available and scope will expand in the future with further validation. Why? Because already some classes of toxic substances have been tested and validated. With more research, there will be all the other groups of classes of toxic agents also will be tested, and it is possible that the scope of the current methods will expand. However, it may not support total replacement of the animal model for at least for the toxicology testing, because one, it is mainly for surface acting agents, and currently there is no method to mimic the xenobiotic metabolism that all the foreign toxic substances undergo after entering the human body. So in conclusion, all the alternatives to animal experiment that we have today will address the issue of reduce. At this stage, it is very difficult to say all the alternatives are going to totally replace. It may be able to replace one particular test, but it is not going to replace the entire panel of toxicology testing. Now, as I showed you in one of my previous slides, let me go back to my original slide where we began with all the panel of toxicology testing. The common panel itself is very elaborate, and we will never be able to replace all the tests. Maybe one or two substances, one or two tests may be replaced. See whether we are going to do acute toxicity testing by using alternative methods, repeated dose toxicology testing by alternative methods, pharmacokinetics, toxicokinetics can we do with alternative methods? Can we do neurotoxicity? Can we mimic a blood-brain barrier? So there are challenges when we come to substances that have to reach organs through various intermediary steps. So currently we are not at a point where we can predict that in the near future there will be a total replacement. But however, some of the particular tests may be replaced, but the total panel of toxicology testing will not be replaced by alternative methods. However, these individual replacements are going to reduce the number of animal usage. Again, even if we are only, if we are not going to use it for all the classes of agents, only for few classes, that will still reduce the number of animals being used. So we can conclude, the overall conclusion is, yes, there are some alternative methods based on cell cultures and in vitro biochemical reactions. And all these are going to help us reduce the use of animals in experiment. But at this stage, probably for next 1 decade, we cannot predict that there will be a total replacement of animals in toxicology testing. So this is my final conclusion, is my personal opinion based on the review of the status of all the alternative procedures that are available. With that, I come to the end of my presentation. Thank you very much. If there are any questions, I would be happy to take some questions.
Sharad, before we go to the questions, we would like to thank you for giving this informative session. And you also updated all of us with alternatives to animal experiments. And also the new avenues which you have highlighted during your talk may be very useful for the new young researchers or our postgraduate students who have joined us. And also how to go about it and exploring these techniques instead of animal experiments has been a new thing for us also, because most of the time we would always think of animal experiments, and as you told, there's always an alternative, and we were not much aware of these alternatives. Sharath, you have given us a lot of input about that, so we like to thank you for that. And we request the participants to post the questions, if any.
If we see the chat box, so far we have not got any questions. Anybody who would like to raise their hands before we have some queries to ask, sir? So first platform, we would like to give it to the participants for the queries. So since we are not getting any queries from participants, our organizers have got 1 or 2 questions for you, Sharath, if you don't mind.
Yes, ma'am. Please
Okay.
go ahead.
Okay, okay.
Sir,
Yes, go ahead, please.
as this is an era of artificial intelligence, is there any role of artificial intelligence to find out newer methods as an alternative for animal experimentation, sir?
Yes, in fact, I did not present that in a big way. I just skimmed through that one, so probably I could not press the message there. So is my slide visible here, skin sensitization?
Yes, sir.
So here, see, if you see adverse outcome pathways, if you know what are the pathways for a given category of toxic agent, we will be able to use artificial intelligence here. So what the whole idea here is, see, you know what are pathways, like the way biochemical pathways are there inside the cells, also very complex pathways are there. But if you can know, these are the 10 pathways that are related to toxic substance. If I give this particular toxic substance to a cell, this particular pathway will be activated, or one of the 10 pathways will be activated. If I know that, then we will be able to apply artificial intelligence. Before we go to artificial intelligence, we need to create the database. So that is why there was some effort almost a decade ago to do what is something called as a human toxome project. I want to study the entire taxon. I am not interested in studying only the this particular toxin or that particular toxin. I want to take one particular toxin as a model, and I want to know what are all the pathways that will be activated by this particular category or a class of toxins. So we can only go to that pathway and check. Now, one thing you will see in whatever tests I mentioned now, this particular point probably I should have highlighted. There is a specificity in all the alternative methods also. We are looking for carnal cells, we are looking for skin cells, we are looking for skin proteins. There is a specificity. When we do animal model, what we are doing is we go by the principles of physiological reaction. Animal will respond. We are not trying to check whether this particular compound is going to cause skin toxicity, whether this particular compound is going to cause liver toxicity. We are not trying to look for the target. We are asking the animal to respond. We just let the animal do whatever it wants, and then we do a overall screen to see, oh, what all happened. So animal study is kind of a passive study, so to say. So artificial intelligence is definitely the future, but to reach there, there is a lot more to be done in terms of research, particularly in combination of toxicology and molecular biology. Now, both these areas are worked separately, but we need to have a synergy in the sense we need to know this particular toxin will cause this particular pathway. If that database of pathways are there, yes, artificial intelligence can play a role.
Thank you, sir. One of the participants in our chat box has raised his hand.
Yeah.
Mr.
Please go
Rajkumar,
ahead, sir.
kindly unmute yourself and ask the question, sir. Sir, he has asked how we can test the toxicity or allergic reaction of allergen or inhaled. Okay. Inhaled allergen.
I'll just repeat the question, Sharath. How can we test the toxicity or allergic reaction of an allergen which has been inhaled?
Yes. So this question, because I presented only one method, which is a monocyte activation, and of course I presented the skin interaction method. So then again, we have to point out here that we are looking at specific targets. So what I presented was only, there's a correct question, that what I presented was only a skin contact experiment. in a simple way when you can say skin contact experiment. Likewise, for every tissue, every category, route of administration or route of contact, we need to validate this method. And that was my summary in the end, that future scope will increase with expansion of the validation, and that is where the scope of research is. Because as academicians, mostly our biggest challenge is where do we do all our research projects? The students have to do, young teachers have to do for promotion. So these are all some interesting questions that people have to take up to validate this method. As far as current standard goes, currently there may not be any method to validate, aerosol based allergens. We have only direct skin contact based allergen method. So methods have to be developed which can address the new unexplored or the alternative or not alternative pathways, the other routes of allergic interactions also.
Rajkumar, I think sir has answered your question because, yeah. Thank you, Rajkumar. There's one more question, Sharath.
Sir, a question is, can we use or trust 3D or 4D printing in producing alternatives to animal experimental models?
Can you repeat the first part of the question? Can we use
or trust 3D and 4D printing technology in producing alternatives to animal experimentation?
To some extent, yes. So overall, I told you alternatives can only be some solution. It is not going to be a total solution, at least in the near future, because we are currently building all these possibilities. Some possibilities are there. 3D is definitely possible and it is playing a role, because if you see two examples that I showed you, reconstructed epidermis and reconstructed corneal epithelium, both are three-dimensional. And I showed you the first limitation there. Cell cultures traditionally, when we are doing cell cultures, we are doing almost for last 50 years in our cell biology subject. We are doing cell culture almost for 50 years now. But it is only in last 10 years some possibilities are opened up for tissue culture. Cell culture means cells grow only in a single layer. Tissue culture means they pile up and they form a multi-layered tissue. Now, the problem of forming a multi-layered tissue is aeration. In the artificial reconstructed epithelium, we do not have aeration. So with that, all the other problems are there. How do you deliver the nutrition? How do you support gaseous exchange? So it is this place where 3D experiment not only has some role, definitely that is the big 3D models, because using those 3D models or 3D technology, you can also create artificial channels for aeration, artificial channels for nutrition supply. With those channels, probably it will be eager to engineer complex tissues, and also we may reach a point where we can reconstruct using normal cells obtained from patients. We may be able to reconstruct organs also, and definitely that is a going to happen thing. So lot of prospects there.
Thank you, sir, for solving so many queries. Now, I request all the participants to turn on their camera so that we can have a photo session. Yes.
Next.
Okay. Thank you all for turning on your camera. Now, I request all the participants either kindly scan the QR code displayed here or click on the link shared in the chat box, and please give your valuable feedback on this interactive session. And I request all the participants to fill the form, as a certificate will be issued only after completion of the feedback form. Yes, me. Now, uh, thank you. It is my privilege to propose the vote of thanks. I, Dr. Arun Hebbar Jayan, on behalf of Department of Pharmacology, Sri Devarajaras Medical College, extend my immense thanks to Dr. Sharad for delivering talk on alternatives to animal experiments. I thank our in-charge Vice Chancellor and Registrar, Dr. DVLN Prasad Sir, for his support. I wish to express my gratitude to our Principal, Dr. Prabhakar Sir, and Medical Superintendent, Dr. Krishnappa Sir, for their encouragement. I thank the management for their continuous support in conducting this event. I also would like to thank all the participants for attending and participating actively in the webinar. I take this opportunity to thank all the faculty, IT department and non-teaching staff of Department of Pharmacology for their help and support. Once again, I thank one and all for choosing to spend their day with us. Thank you. Thank you all for participating and for your active participation. We wish to see you all in future events. Have a great day. Now you can all log out.